and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with virologists, students and postdocs that belong to the American Society for Virology so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On May 31st, 2022, we talked with Isabel Lewis, a graduate student at the Pirbright Institute in Surrey in the United Kingdom. She studied bioveterinary science at the University of Lincoln in the UK, and she is investigating several virulence factors of lumpy skin disease virus, a poxvirus of cattle that has recently emerged from sub-Saharan Africa. Thank you for talking with us today. Why don't you tell us a little bit about yourself?
Well, firstly, thank you for having me. It's really an honor to be able to share my work and how I got into virology. So my name is Isabel Lewis. I work at the Pirbright Institute. I'm a final year PhD student, and my PhD has been in collaboration with the Friedrich Institute, so they're in Germany, and also the Royal Veterinary College in London. So I have a background in bioveterinary science. That was my undergrad. And then I since found my love for virology during my undergrad mainly, and has managed to get into my PhD position where I am now.
Great. And how did you become interested in science, or way back when, middle school, high school? What started it for you?
So I always enjoyed science at school, but I was much more of a sporty person. I thought I'd be a PE teacher. That was going to be my life. And I was lucky enough to go on a volunteering expedition in Kenya. So I was 16. And I think the interactions with wildlife and nature made me understand that I actually have a deeper love for animals than sport. So for me, it was my love for animals which changed my career trajectory. So instead of doing A levels, I did a BTEC qualification, so that's a more hands-on qualification in animal management. And from there, I realized I wasn't a big fan of certain aspects of like animal management. I cared more for the science. So then from there onwards, I was like, well, how can I use my love for animals and science and use that to make a career out of it, basically. And then I looked at different degrees, and bioveterinary science was the one that shot out to me because I didn't want to be a vet. I knew that. I knew the hard hours and the long days wasn't for me. And then I saw that bioveterinary science was very much kind of like a biomedical science for humans, but just focused more on animals rather than zoology, which is more conservation and welfare based, whereas I was much more interested in the hardcore science. And then from there onwards, I loved the virology lectures. The infection sciences modules were my favorite. And the virology, how you have so many different viruses that can cause so many different diseases, and they come in much different shapes and sizes. And the names were also amusing, I have to admit. We pick a good array of names for viruses. And yeah, that was what's really stemmed my career, is my love of animals, then found my love for viruses, and I combined the two together.
Right. And how did you find sort of your particular lab or your institution? Can you describe to us sort of like how that happened?
So luckily, I did my dissertation with my supervisor at the University of Lincoln, so during my bioveterinary science degree, and they had previously worked at the Pirbright Institute where I am now. So they knew that it fit in with what I was really interested in research-wise, and they really helped support me in my applications. And I was very lucky. The first PhD application that I applied for, I got. So I was very much very naive and applied for one that I just thought sounded amazing. So it was investigating the virulence factors of lumpy skin disease virus, a virus I'd never heard of before. So I knew it was going to be fun to research a completely new different field, and a field where there's lots of gaps in the knowledge. So I knew it'd be an interesting PhD to do. And my lab group are amazing. So I have a really strong, fierce boss. So Dr. Pitbeard is honestly amazing. If you've ever met her, I would highly recommend to meet her. And she's made our lab such a positive environment. So we are I'd say a biggish lab. We have 3 PhD students at the minute, and we have I think, 4 postdocs as well. So it's a really good array of both students and postdocs, and we all work on different elements of lumpy skin disease virus, and then we can combine and interlink and see how they all relate and be able to make progress in further understanding the virus and the disease.
Cool. And lumpy skin disease virus. I have to say, I have not heard of that one before, even though I've been in virology for almost 30 years now. Can you tell us a little bit more about it? And then maybe talk about your PhD thesis work, sort of the big picture, but also sort of some of the techniques that you use, like how do you actually do your studies?
So you're not the first to have not heard of lumpy skin disease virus. So it's a poxvirus, so part of the poxviridae family. Large DNA virus, so you can't see it on one screen. It's not 8 segments of a little virus that you can work with, which makes it more interesting in my opinion. But lumpy skin disease virus causes lumpy skin disease. Again, we're not very imaginative with the names. It does what it says on the tin. And this is a disease that infects cattle and water buffalo. So it's a important pathogen that affects obviously cattle livestock. And it was originally endemic in sub-Saharan Africa, and has since spread over the last decade into the Middle East, Eastern Asia, and Europe. So it's, it's quite a neglected pathogen despite this rapid spread of the disease. And the disease causes your characteristics like pox-like lesions on the skin. It causes a reduction in milk yield for cattle. It causes weight loss, as well as like ocular nasal discharge. So you can imagine the economic impact this has on the farmer. And countries have had to obviously spend a lot of money for control methods, and the main control methods are live attenuated vaccines. So there's an array of different commercially available vaccines, and they have a strong lasting immunity and they have good efficacy. But this has not prevented the disease from spreading still. We've still seen this big spread, and currently it was noted in Indonesia last in March this year. So now countries like Australia and New Zealand are obviously very aware of this virus, and they know what the virus can do to their cattle populations. Obviously, they're big exporters of cattle products. So that shows more research is needed to be done in the prevention of the disease and to understand the virus more, considering other pox viruses such as vaccinia virus, ectromelia virus. I mean, there are obviously monkeypox viruses in the news at the minute. There is little known about lumpy skin disease virus. And my project fits into that by trying to understand the virulence factors of lumpy skin disease virus. So what genes are encoding proteins that can block and inhibit the innate immune response? So that's where my project kind of fits in. And we're hypothesizing by deleting these virulence factors, we can generate a rationally designed new vaccine, basically. So part of my project that I'll present at ASV is looking at lumpy skin disease virus effect on the type 1 interferon pathway. So we're looking at an extracellular encoded protein that can bind to type 1 interferon and block that pathway. And type 1 interferon is important because it induces the antiviral immune response, which is known to interfere with viral replication and dissemination. So poxviruses, being the huge DNA viruses they are, have encoded a multitude of immune evasion proteins to block type 1 interferon signaling, and nothing has been discovered yet in lumpy skin disease virus. So that was one avenue we've taken. And another is looking at VBCL2 proteins. So they're virally encoded BCL2 proteins, which are shown to play a role in apoptosis. And interestingly, vaccinia virus, another pox virus, encodes a multitude of these VBCL2 proteins that block NF-kappa B signaling. So we were looking to see if LSDV, so lumpy skin disease virus, has also these orthologs that can block NF-kappa B signaling as well. So 2 different pathways, very interesting, both of them. And yeah, hopefully we can then take this work forward. So what we've done is looking in vitro first. We have seen if these proteins, both VBCL2 and potentially a type 1 interferon decoy receptor, can actually block these 2 different pathways. And then we are going to take this further forward. Well, I say we, unfortunately, I'm finishing up my PhD. I'm in that fun phase of writing the thesis. But this work's going to be taken forward with these deletion viruses that we've created, definitely in the type 1 interferon part of my work, to see whether this creates an attenuated response in vivo. So we have an animal model at the Pirbright Institute to study that. And then also to see whether this virus that has this gene removed can actually be a vaccine candidate as well. So that's future work. But in my project, I have focused on characterizing the proteins in vitro. That's what I've done. So using cell-based assays such as dual luciferase reporter assays, ELISA-based assays as well, and also immunofluorescence, because that is my favorite technique.
Great. And when you say that you're making deletion viruses, can you describe how you actually engineer or sort of change a DNA virus? Obviously, it's not as simple as sort of a 7 kb RNA virus. So how do you do that?
No, our virus is about 150 kb. So it's a lot different. You can't see it on one screen. So what we have to use for poxviruses is homologous recombination. So we will infect our cells with our virus, so LSDV in this case, and then we'll transfect them cells with a plasmid which encodes our protein of interest to remove. So what I have is basically a marker plasmid. So I have an EGFP tag. You can have different tags. And then I have flanking regions either side of the gene I want to replace. So then we get recombination in vitro. And then what we've since used is single cell sorting to isolate the EGFP expressing cells to the non. And with the cells that we use as well, they have very low transfection efficiency. So we need this technique of single cell sorting to identify these EGFP colonies. And then we take them forward by limiting dilutions to make sure it's not contaminated with any of the parental virus. And then we have taken that further forward with next generation sequencing to confirm that the deletion is truly deleted. And then we've taken this markered virus, so we've now got an EGFP tagged virus which lacks our gene of interest, and we've taken that forward to remove the EGFP by using LOXP sites that flank this EGFP region. And then we passage this virus into MDBK cells, so that's the cell line that we use for lumpy skin disease research, and we infect them cells which express CRE to then excise the LOXP sites to remove that EGFP marker. And the reason we wanted a markerless virus was to take forward into the animal trials, because we don't know if EGFP would have any beneficial or negative effects, but we'd rather avoid that if we can. And luckily that's all worked using the same method that we use to generate the EGFP virus, but instead using obviously the EGFP as the parental virus, and then using that single cell sorting to remove the EGFP population from our markerless population.
Right. And one question I had is for this pox virus and maybe others, how species restricted are there? So is this a disease, a virus that infects different types of cattle, but can actually periodically infect other livestock or humans? What is known about that?
So lumpy skin disease virus is in the Capripox genus, and they're known for being quite narrow host range. So lumpy skin disease only infects lumpy skin disease virus only infects cattle and water buffalo. So very narrow host range compared to other well-known pox viruses that can infect a much broader range, which makes it easier to study in some regard. But we can't use a mouse model to study this bovine disease, unfortunately. And with sheeppox and goatpox, which is another members of the Capripox virus genus, it's in the name, they infect either sheep or goats. So they're very much a narrow host range from these viruses.
And is the receptor known for the lumpy skin disease virus?
No. So there's no receptor that's been linked to lumpy skin disease virus as of yet. But I think pox viruses mainly enter cells via phagocytosis and macropinocytosis. Okay. So it's not, I don't think they've been linked to a receptor mediated entry, though I'm sure I could be quoted wrong. There are receptors that are involved.
Yeah, in that process.
We just don't know them yet. Yeah.
Okay, cool. So you were saying that you're at the end of your PhD, so you're wrapping up. What are your next steps? What are you thinking about doing next then?
I was quite open to a lot of different careers. So obviously some people like to remain in academia, some go to industry, some change careers altogether. I really wanted to enter lecturing. So I said this from day one. In my interview, I remember saying I wanted to lecture, and I've been lucky enough to be offered a lecturing position at an agricultural and sports-based university college in England. So I get to stay in my home country. So my mum is very happy. And I've obviously now thinking of long term, it's a permanent job. And that was the thing that turned me off a bit about staying in academia and postdoc positions, was the consistent moving around. As much as there'd be great opportunities in great lab groups, I was much more looking for a permanent career choice.
Cool. And when you say a lectureship, what does that consist of?
Yes, so it's going to be an animal science lecture role. So I'll be working on the animal health and disease modules, potentially microbiology modules as well as anatomy and physiology. So it'll be predominantly a teaching role, but I'll still be able to run some research, which would be very interesting.
Oh, cool.
I'm hoping to run some interesting projects with the students. It's going to be exciting to hopefully encourage the next generation of scientists into the field. I mean, who knows? I'll hopefully have the next Nobel Prize winner. You never know.
Right. And I guess thinking about that, is there a particular virus or sort of type of experiment that you would like to continue, research that you would like to continue in your own lab, as it were?
I'd love to stay with viruses. However, I've got to consider the facilities I'm at. So at Pirbright, we have the high containment facilities. So I don't have that in this new job. So it's definitely going to be a learning curve of what I can do within that institute and what I want to do. So I'm hoping to work on potentially poxviruses, because they're still important in the UK, and I still get my poxviruses, which would be great. But I'm looking to expand into other diseases as well and other pathogens. So I'm looking at Toxoplasma gondii, it's a really interesting prevalent, obviously, parasite. So we shall see.
Okay. And does it have to be diseases of agriculture or livestock, or can it be pretty much anything that you want to study?
It could be pretty much anything by the sounds of the new university I'm going for. My love is for livestock diseases, so I will probably stay in that field. I did research focusing on avian diseases, so it'd be nice to go back potentially into that area, as well as bovine, caprine and ovine diseases.
Right. And then I guess just finishing up, it's sort of an interesting time to be a virologist. What has the last 2 years of the COVID pandemic been like for you? How has it affected you in work, professionally, but also personally?
Well, luckily I got my first year of my PhD done, so I did have some normality within my PhD. But I was actually at the Virulence Institute in Germany, March 2020, and I got an email from different people saying, I think you should come home, because I planned to come home a week after these emails, and it was all starting to get a bit more serious. So I came home early, and then a couple of days later, England went into lockdown, and Germany stopped flights going from there to the UK. So I was very lucky to get back to my home at least. And my partner was the unlucky one in this, because we'd been not together too long, and I asked to live with him after saying we'd have a long distance relationship. I came back to the country and was like, please take me. So we did move in together, and he's a teacher. So he was doing online lectures and sessions, and I'm there trying to do some in silico work instead of, obviously I couldn't work in the lab. So I decided to go down that avenue while I could. And I got very distracted listening to history classes. So it was fun. I got to learn a lot of different things. So if you ask me now, I wouldn't know. All I know is lumpy skin disease virus at this moment in time. And it was hard to not be in work in the lab, but I think it was harder going back, because it was such a long time that you weren't there. It was harder going back and seeing lots of people and trying to know how to interact with humans again in society. Though I was lucky enough in the pandemic, the first lockdown in Britain, I got to work in a hospital lab. So I got to help run the PCRs there and managed to contribute and help and use my skills, thankfully, in this pandemic. So that was a nice break from working from home all the time. But we did get to go back to work not too long. I think it was May 2020 we got back in the labs. Obviously, there was a lot of restrictions. So generating the deletion viruses, I was working with a postdoc and trying to be able to train someone socially distanced was a challenge. And we managed to work through that. Well, obviously we got successful deletion viruses. So we did manage to, go through them hurdles. But now it's nice to see everything kind of being more back together. Though there's still things that are changed and they've stayed that way. So Teams meetings are much more common now. Like when we present internal seminars and external seminars, I think there's a high majority of people that still like the Teams rather than the in-person, whether that's just because you could sit at your desk, you can eat a roll and no one's seeing you get sauce on your face or, other reasons. But yeah, there are definitely benefits that have happened from the pandemic as well as negatives. And I think when you look back at the human kindness that we did see around the globe, there were good things in this pandemic.
I guess I didn't clarify. So when you talk about using animal models, what kind of a model is it?
So we have an animal model, and it's similar to other groups in Belgium and FLI, where they inoculate cattle with a needle bolus in either intravenously or intradermally. So we use that to study the LSTV in cattle because we can't use another model. We can't use another animal model just to its narrow host range.
And is that like high containment?
Yes, so it's in high containment animal facilities. So we take all the samples into our high containment lab to then work on them because obviously we don't want to release LSTV into the naive population.
All right. Well, thanks so much for talking with us. It was very interesting to talk to you about your research, and we look forward to hearing more about it at ASV this summer.
Thank you for having me. Thank you very much, Larissa.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.